about my research regarding proteins involved in aging. So as you all know, what got me here was a rat video, and after a lot of discussion with my rat mentor, we decided that it kind of misrepresents the lab. So I brought her here today to kind of clarify some things. Hey, Dr. King, are you there?
Yeah, I'm here too.
Good.
Let's break it down.
Irrigation facing the problem, work the racing, we'll solve it with the silence, with some patience, and climbing, one of the greatest, climbing trainers, over the time of the day, stay alive, and just be praying, we find evidence, what you think you went.
Stop the generations of nations, save this aging nation from impaired cognition, cause of
Destroy the condition, that's separate the condition. Seems like these guys forgetting everything with your permission. I will explode this now, so pay attention, don't write me down. When we listen to our FGF 23 journey that we have found so profound. You're learning what we know now is clocked out.
No, it's clocked out. You should know why. That's what our findings is all about.
For real, I'm teaching this crowd. You're really gonna correct me right now. I feel like I should start my lecture and kind of stop the speech. Just shut it down.
I'm sorry about her. Anyway, just continuing forward. Well, I'd like to ask everybody a question. What do you think the greatest risk factor for developing neurodegenerative disease is? That's right, it's Jersey Shore, but we can't seem to get rid of that one. So science focuses instead on the second leading cause, aging. As you can see here, as age increases, so too does the prevalence of developing dementia, which has led science to many questions like how do we slow aging to better prevent neurodegenerative disease, or disease in general. So our lab focuses on the aging brain to help answer these questions, specifically two proteins, FGF23 and klotho. And my focus in the lab is to determine if klotho-deficient premature aging is caused by the lack of fibroblast growth factor FGF23 signaling. It's a mouthful, so I like to break it down a little bit for everyone. So as I mentioned in my rap, and actually in both raps, klotho is the main focus of our lab, and it also happens to be the name of the Greek fate that was in charge of spinning the thread to everybody's life. You know, the one from Hercules. So it only makes sense that this protein that we work with is called klotho, because mice missing this protein die much more rapidly, as you can see here by the survival curve, compared to the wild type. And when given just a little bit more klotho, these mice live up to 30% more longer lives. It's remarkable that this one protein can alter lifespan this way, but what's even more profound is that when changing this one protein within the brain, you can rapidly and significantly alter hippocampal function, or brain function, specifically within the hippocampus. And knowing this, we tested klotho knockout mice for the ability of them to recognize that an object has been moved just 1 centimeter over. And what we find is that these klotho knockout mice fail this task every single time, developing cognitive impairment within 1 week. So how does klotho function in the brain to regulate cognition in such a way? Well, in these klotho knockout mice, we specifically study the hippocampal neurons, and in these neurons, we test them for the ability of one neuron to communicate with another neuron, synaptic transmission. And in testing these klotho knockout mice, we find that there's both enhanced pre- and enhanced post-synaptic functioning, which is kind of the opposite of what you would expect comes along with cognitive impairment, but trust me, these mice are really, really poor off. In addition, when looking in the hippocampus for adult neurogenesis, measuring cell populations ranging from stem cell to immature neuron, we find that in the klotho knockout mice, there are fewer cell types at every developmental stage, which is kind of meaning that we are defining the premature hippocampal neurogenic niche, which is really interesting. But how is klotho doing all of this? So klotho is both a secreted and a transmembrane protein that exists within the brain and the body, but virtually all of the science that has been done on it is specifically within the kidneys, where a lack of klotho is absolutely deadly. The way it works is that FGF23, see we're getting there, binds to klotho in the kidneys, allowing them to bind to FGF receptor. If this mechanism functions properly, you can excrete excess phosphate from the blood. However, if one thing is messed up, you die from multisystem organ failure. You know, no big deal. In addition to this, the FGF23 and the klotho knockouts are phenotypically similar, so this has just led science to basically put these two in the same basket. They have to work together. No one's ever really looked in the brain, though. That is until us. So, in testing these FGF23 knockout mice for the same things that we tested the klotho knockout mice, synaptic transmission, adult neurogenesis, we find that in testing the hippocampal synaptic transmission, there is no change in either pre or post-synaptic functioning, unlike the klotho knockouts. And in adult neurogenesis, when looking at the cell populations from stem to immature, we see no change in cell population or cell count numbers at any point along development. So this is pretty remarkable because they still have cognitive impairment when testing. These FGF23 knockout mice still have cognitive impairment when we test them with novel object location. So this is very strange because neither adult neurogenesis nor synaptic plasticity underlie this mechanism, so this has led us with many, many questions. The first one is, we kind of know as a scientific community that kidney disease directly leads to secondary cognitive impairment, so perhaps what we're seeing in these FGF23 knockout mice is what the brain looks like after developing kidney disease. This would be intriguing, but would even be more fascinating, or any other word I haven't used for that, would be if FGF23 and Klotho have independent, unique functions within the brain that could help our lab and the scientific community better understand aging as a whole. Thank you, and I'd like to say thank you to everyone that's currently in the lab, previously in the lab, or futurely in the lab. I'd also like to say thanks to everyone that helped me with these videos, the YouTube viewers, and basically everyone that helped put this on. Thank you so much.